Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Aisha Rashid, Senior Life Science Journalist at Xtalks.com, and today we're going to be talking about innovation in the rapidly evolving field of cell therapy. To discuss the topic, we have a very special guest on the show. Joining us, we have Dr. Ian Nisbet, who is the Chief Operating Officer and co-founder of Cartherics, a biotechnology company developing cell therapies for hard-to-treat diseases, including cancer and endometriosis. The company is based in Australia, and Dr. Nisbet also acts as a director or advisor to several Australian and U.S. biotechnology companies. Dr. Nisbet has more than 35 years of leadership in biotech and experience in bringing two FDA-approved oncology drugs to market. He offers unique insights into how the next generation of cell therapies could transform patient care. On the show, we'll learn about Cartherics' proprietary platform for generating an effectively unlimited supply of CAR-NK cells, the challenges and opportunities in developing treatments for underserved diseases, and what's next for the cell therapy industry worldwide. To learn more about all of this and more, let's turn to Dr. Nisbet now. Thank you so much for being on the show with us today.
It's a pleasure, Aisha.
So, Dr. Nisbet, I know you've asked me to call you Ian. I don't even know where to begin because you've had such a remarkable career spanning more than 3 decades in biotech, as I mentioned. Can you share a bit about your journey? And I know this won't do justice to it, just the couple of minutes on the podcast to introduce yourself, but, really from developing FDA-approved oncology drugs to now co-founding Cartherics and leading such wonderful work in the space of cell therapy. Could you tell us a bit about how you got into the field, specifically from oncology to cell therapy? Obviously, in oncology, cell and gene therapies are the cornerstone in that space, but we'd love to learn more about it from you.
Yeah, sure. I started my career as a microbiologist a long time ago, and a research assistant at Melbourne University. Moved to Australia's largest pharmaceutical company, CSL Limited, way back in probably the mid to late 70s, and grew with that company. It started off life as a government-owned organization, and eventually has become a international pharmaceutical company. So I was there at a time when it was a much smaller company, but I had the opportunity to grow with that company, learn about the pharmaceutical industry, learn about project management, learn about licensing and so on. And so I was at CSL for nearly 20 years, working in a whole range of different roles, and then had the good fortune to be tapped on the shoulder and asked if I would be interested in going across to work in Boston at Millennium Pharmaceuticals, which was one of the early genomics companies. And I had known some of the people there. Actually, I'd visited Millennium when I was working at CSL, and got to know a few people there, and they asked me to come across and work with them. So I had the really good fortune of spending some incredibly exciting years in the late 90s and early 2000s at Millennium. And that's where I really got involved heavily in oncology, because oncology was one of Millennium's franchise areas. And one of the companies that we acquired had an oncology drug in the development pipeline. It was a drug, it was originally called PS341. It became Velcade, and that was the drug that Millennium got approved by the FDA for the treatment of multiple myeloma back in the early 2000s. And we ultimately launched that drug ourselves in the United States and partnered with Janssen J&J, who launched it in the rest of the world. And I had the really, really good fortune of being the project leader when we got that drug approved. So it was just a tremendously exciting time. And what it did for me then, particularly with the Velcade experience, was just really appreciate just how profoundly you can change patients' lives with a drug like Velcade. Eventually, it became a multi-billion-dollar drug. It was one of the first drugs approved in multiple myeloma for decades back in the early 2000s. And since then, there's been a lot of new drugs developed for multiple myeloma, but it was one of the first ones that really showed that you could change the course of that disease. And I'm sure there are still people alive today that are alive because of Velcade. And so that experience was tremendously empowering, and wanted me to do it again and again, and really was the basis for my journey in oncology. Since then, I have worked with a number, mainly oncology companies. Came back to Australia, worked with smaller companies, founded a couple of companies myself, and primarily focused in the oncology space because that's where I'd had the experience with Velcade, and subsequently another drug called Cinribo, which was developed by Chemgenics, where I worked for a number of years. So oncology has been sort of the key to my career for the past 25 years or so, and ultimately led to the founding of Cartherics with several other founders, and that happened because I was aware of what was happening with CAR T cells. I wasn't involved in the space when that field was emerging, but what happened was Alan Trounson, who is the CEO at Cartherics, he was president of the California Institute of Regenerative Medicine back in the sort of 2007 to 2014 sort of period, and he had been funding CAR T technology through CIRM, and he came back to Australia and decided he wanted to create a company in the space, and it just so happened that he had previously worked with a guy by the name of Bob Moses on commercializing in vitro fertilization, and I'd worked with Bob at CSL, and so Bob connected me to Alan, and we decided that he had the stem cell knowledge, I had the oncology experience. We had Richard Boyd, immunologist, Bob, commercial person, and Peter Hudson, an antibody expert, and we decided that we would create Cartherics to leverage our skills and experience. And so that's how I became involved with cell therapy. So it wasn't that I had any particular background in cell therapies at the time we founded Cartherics, but just that I had that oncology background and the connections. And it just sort of shows you that the power of networks, that it was basically via knowing people who knew people that we got together and created Cartherics. So that was about a decade ago. So it's been an interesting journey to get to this point. Over the period of my career at CSL, at Millennium, at some other biotechs that I've worked in, as well as Cartherics, I have worked on small molecule drugs, I've worked on antibody drugs, and now I'm working on cell therapies. And each has their unique sort of benefits and challenges, and they each have their place to play in the treatment of diseases like oncology.
It's wonderful. It just feels like all of the right ingredients, so to say, and elements coming together for a perfect storm almost. With all of your background, of course, in oncology and being kind of the poster child or poster field for cell therapies, I think definitely the right people coming together to make things happen at Cartherics. And in terms of, we talk about the missions and goals of a company, but what really is, of course, the mission is to help patients at the end of the day, for most companies, I would imagine. What is Cartherics focused on primarily? And I know that also I was reading that it's just also you're also working in the area of endometriosis. So that's very interesting in terms of oncology and gynecologic conditions. So did those two seemingly separate fields, but I'm sure there are some commonalities in terms of principles and all of that concepts. But I'm very interested to hear how the company is focused on these two areas.
Yeah, so it's an interesting connection, that one. So we started when we started Cartherics, like pretty much every immune cell therapy company, we were focused on oncology. That's where the original CAR T results came from, and the results from the first CAR T studies were just amazing. So I've worked in oncology a long time, and generally we think about advances in incremental terms. An extra few months of life. Uh, Extra 10% response rates or whatever. We very rarely see huge differences associated with a particular treatment. And what we saw with the original CAR T results was in rare blood cancers, and it has been limited to rare blood cancers up until now. But we saw response rates approaching 100%. We saw um, people who had failed all prior treatments, who had effectively no options, be treated and have complete responses. And some of those people are still alive 10 years later. And so with that type of therapy, you could start thinking about cures to cancer rather than just extending life. Previously, really, we thought about incremental advances. No one talked about cures. And so the first results with CAR T cells was just so profound that it really caught people's imagination and our imagination at Cartherics. So we decided we wanted to work in cancer. That was where the huge need was, and where where the initial results were. And what we decided was that rather than look at rare blood cancers, which is where most people were focused, we would we would go for the bigger need, the bigger hurdle, and that was with solid tumors. And it just so happened that the target that we chose, a tumor target called TAG72, that we decided to work on for our first CAR T. We started on CAR Ts, now we're working on natural killer cells, but CAR NK cells. So our first target there was a molecule called TAG72. And it just so happened that that marker is expressed on a whole range of different tumor types, so-called adenocarcinomas. But the one tumor type where it is expressed by the vast majority of tumors, 90% plus, is ovarian cancer. So we decided that ovarian cancer would be our first target because of the nature of TAG72 expression, but also because there was a huge unmet need there. And it was one of the diseases where there wasn't so much competition. So we thought, for us, that was a good disease to go after. So ovarian cancer was our first target. And it will be the first indication that we go into the clinic with our CAR-NK product, CTH401. But it's so immediately we had a focus on gynecological cancers. And it just so happened that Alan, because of his background in vitro fertilization, he has a tremendous network in the gynecological community. And he went to a conference and he was talking to some of his colleagues, and they said, well, gee, I wonder if that would work in endometriosis, because there's, just a huge unmet need there. And so what we started doing was looking at the biology of endometriosis, and in particular, the expression of different markers. And what we found was there is some evidence for TAG72, our ovarian cancer target, being expressed in late stage endometriosis. And then even further than that, a publication came out last year in JAMA saying that, or demonstrating that there was a definite link between endometriosis and ovarian cancer. So patients with endometriosis, particularly late stage severe endometriosis, are at much, much higher risk of developing ovarian cancer than women without endometriosis. So there's clearly a biological link there. And so that gave us encouragement that, we're actually maybe targeting markers that are involved in both diseases. And then even further than that, so TAG72 is our initial tumor target. We also became interested through a relationship with Ohio State University in tissue factor as another cancer target. And it turns out that tissue factor is expressed in the vascular endothelium of endometriosis tissue, and it's expressed pretty much on all endometrial tissue. So we have another biological link there. So partly actually, it's mostly serendipitous that we actually started working in ovarian cancer, and then by virtue of the targets that we were going after and this biological link between endometriosis and ovarian cancer, we have moved to say, well, we really should be focusing on both of those diseases. And so what that does is means it sort of repositions the company as focusing primarily on poorly treated women's diseases. So we're sort of repositioning the company now as a women's health focused company. And what that does is it gives us a nice focus, hopefully allows us to get interest from investors and partners who have an interest in women's health, and also differentiates us from other cell therapies companies, because other folks are they're focused in cancer. A lot of them still in the rare hematological tumors, but those companies are also moving into diseases like autoimmune disease by virtue of the targets that they're going after. And so they're diverging down one path, and we're diverging into another path. And that's sort of helpful in terms of, again, differentiating us and basically giving us a story that we put out to potential partners, to potential investors, that is a little different to what they're hearing from other cell therapy companies.
Absolutely. Thank you so much for sharing that. Really a lot of intersectionality, and we talk about, interdisciplinary kind of work there, and the concepts and the targets being identified in, again, from oncology to women's health conditions like endometriosis. So that's so intriguing and really, really interesting, and indeed does set things apart. And in terms of the focus of Cartherics, you mentioned that CAR NK cells is your area of focus at the company. And so could you talk to us a bit more about that, and how would you consider those to be next generation CAR T cell therapy, or just a different aspect or a different type of CAR T cell? Or how is that evolution happening?
So it's a variation in the field. So people started originally working with CAR T cells, and T cells are they're part they're an important part of the immune system. They are very, very aggressive at killing cells. That's their job, and they have obviously have a very, very clear role to play in the treatment of diseases like the rare blood cancers that they're approved for, and hopefully we'll see some ultimately approved in the treatment of solid tumors. They are very aggressive cells. They and as a result of that, they have a range of side effects that can be life-threatening. So they produce what's called cytokine storms. So when a CAR T cell attacks a tumor, you get a release of huge amounts of cytokines, particularly interleukin-6, and they can cause basically ultimately death. There are a number of people who died as a result of CAR T treatment. So because they're so aggressive, you get these really strong induced immune responses, and they can have very, very bad side effects. There's also a neurological symptom or disease associated with CAR T cells called ICANS. So some people have died from that as well. And so T cells, because they are more aggressive, they're more likely to have a place where you really need an aggressive type of cell, and you don't have any other options. So where you have late stage cancer, and you don't have other treatment options, a CAR T cell is a reasonable thing to do. Natural killer cells are a little bit different. So they're part of what's called the innate immune system. So their job is to just be there, kill aberrant cells, tumor cells early on as they arise, viral infected cells and so on. And so they monitor the body and just mop up cells that shouldn't be there. They are more benign than T cells, so they don't cause things like the cytokine storm or cytokine release syndrome. They are not associated with graft versus host disease, which T cells are, and so they are an inherently safer cell type than what T cells are. So you don't in the clinical trials that have been done with CAR T cells and CAR NK cells so far, CAR NK cells have a pretty benign safety profile. So you can put a billion CAR NK cells into a person, and you generally don't see any significant adverse effects. So they provide a safer baseline, and when we think about them for treating particularly non-cancer diseases where safety is going to be important. So when you think about endometriosis, those patients, they have a chronic, very poorly treated disease, but they are not at risk of dying. And so you're not going to be able to treat that patient with a therapy like a CAR T therapy, where there is potential risk of death or severe disease. So you need an inherently safer cell type there. And we think natural killer cell is that type of cell where you can, once you've demonstrated that it is safe, and our plan is we'll demonstrate safety initially in cancer patients. Once we've done that in cancer patients, we can logically move then to earlier stage cancer patients and healthy patients like patients with endometriosis with that cell type. So we think a natural killer cell provides a platform that is a bit more versatile than a T cell platform. And that's not to downgrade the value of T cells. And ultimately, our thinking is that there'll be a place in treatment for a range of diseases, for T cell based products, for NK cell based products, for macrophage based products, another immune cell type, and so on. And each of them will have a place to play in one or more diseases, and probably in one or more stages of different diseases. So again, the earlier stages. So we're thinking for cell-based therapies in cancer, where they're most likely to be most effective, at least in solid tumors, is early on in the disease when there's low tumor burden, and where you can mop up tumor cells, prevent metastatic disease, and so on. And so we're thinking that ultimately our sorts of products will play out as maybe an adjuvant treatment being used at the same time as surgery or something like that. And so ultimately, you need a safe type of cell for that type of patient. So it'll be different cells, I believe, different types of cells for different diseases at different stages of the patient's journey.
Wonderful. Thank you so much for setting the stage and putting all of that into perspective. And do you envision that cell therapies can be used in combination, or is the CAR NK platform that you're developing, is the idea to use that as a monotherapy, or how do you envision that?
Sure. I mean, it's a great question. So cancer is a disease treated by combinations. So generally what you have to do is show single agent activity, and then you start looking at logical combinations of that therapy with other approved therapies. Will be no different. So our first clinical trial will be our lead product, CDH401, as a monotherapy, and in late stage patients. That's just the way you have to develop these things. But ultimately, it would make sense to think about it in combination with the existing treatments. In ovarian cancer, you would think about maybe combining it with checkpoint inhibitors. So there's some evidence for checkpoint inhibitors having a role, not a huge role, but a role to play in ovarian cancer. It would be logical to combine a cell-based treatment with an anti-PD-1 antibody or something like that. Of course, then things start to get expensive, and you have to worry about costs. There's also an approved antibody drug conjugate in ovarian cancer. It targets the folate receptor, so that could be complementary to targeting TAG72 with a natural killer cell. And then, of course, there are targeted therapies as well that you could think about combining. You could also think about combining with chemotherapy, although there you'd have to get the sequencing right, because you obviously can't use chemotherapy at the same time as you introduce a cell therapy and kill off all the cells that you are putting into the patient. So it would be a matter of maybe doing chemotherapy, then cell therapy, or cell therapy, then chemotherapy, and just getting the sequencing sorted out. So there's a lot of work to be done, and that's one of the challenges, particularly for a small development company like Cartherics. You have limited finances, you have a limited number of trials that you can run, and so basically you want to get your product into the clinic, demonstrate safety, demonstrate some evidence of efficacy, and then you want other people to play around, do the different combinations, work with partners, work with oncology groups, cooperative centers and so on, to actually look at ways in which your product can be combined with other ones, because the amount of variations that that need to be tested are huge, and it just can't be done by any one company. And again, as I said before, hopefully, ultimately, in our view, these types of products should be most effective when they're used early on, when there's low tumor burden. And so the idea of combining our cell-based therapies as adjuvant therapies with surgery in early stage treatment and so on would be a logical way to go. But that they require really long clinical trials beyond the scope of a small company. We really rely on the cooperative groups and stuff like that to do those sorts of studies. So developing these products is a journey. It's not you don't do phase one, phase two, phase three, get it approved. It actually is a journey that lasts decades.
Yeah, absolutely. Because you have post marketing, and there's then a whole ecosystem, commercialization that develops around it, a product. So definitely. And as you mentioned, that, it's with limited resources, it becomes difficult on the development side and scaling. So what types of partnerships become important then for a small, mid-sized biotech in order to really push the effective development of a product or candidate?
Yes. Ultimately, where you need the partnerships is for the later stage clinical development and commercialization of the product. So we're no different to most other biotechs who have the objective to get human proof of concept data and then leverage that the initial human data to a partnership with a major player, a pharma or a large biotech, that would then conduct the phase two, phase three studies. I mean, hopefully with this type of product, you would be able to get accelerated approval based on phase two, decent sized phase two data or decent sized phase two study, but then you still need to do the phase three study effectively as a post-marketing study or in association with approval, and then the commercialization. And cell therapy products, they're not simple products to commercialize. You've got to have cold chains and distribution and everything, and even the use of the products in the clinic is not so straightforward. You have to go with to specialized centers who are used to dealing with cell therapy products and so on. So all that sort of work is beyond the scope of a small biotech, and so we, like others, look to get our product into the clinic, demonstrate that we can do that, get safety data, some indicative efficacy data, and then we would look to partner. And because we've got a platform, so our platform is we start with induced pluripotent stem cells, we gene edit those. And fully characterize those gene edited cells, put down a master cell bank, and then we take the cells from the master cell bank and expand them and differentiate them into the final cell type, which is in our case a natural killer cell. But because we have that stem cell derived platform, the iPSC derived platform, we can make multiple gene edits. We can change. We can put different CARs in there, so target different tumor targets. We can knock out different genes that are associated with immune suppression or metabolic function or whatever. And so we can edit those cells to be the starting point for multiple types of products. And then through the differentiation process, we can push those cells to become natural killer cells or T cells or macrophages or whatever. So we have a platform that has multiple potential products. And so we have the flexibility then when we think about partnerships to partner either on a product by product basis, or if we find a really good strategic partner, we can partner the platform as a whole. So we have, from a business development option, we have a number of ways that we can go. But what most partners want to see is the first human data, and so that's what we're pushing for. And so we're very focused on getting that first clinical trial up and running, and then we'll be in a position to start talking with potential partners.
Wonderful. And in terms of the platform, I did read about how the basis is to produce an unlimited supply of these CAR NK cells. And so is that a way to really address the challenges, one of the challenges in cell therapies, to produce and expand your CAR T cells or your CAR NK cells in this case? And so can you talk to us a bit more about the platform, and also connected to that, of course, the challenges today? In your perspective, what are the challenges in developing cell therapies, and what is needed, and where do you see the field heading from where it began, let's say almost a decade ago, where we started seeing those results in patients and products getting into the market, and where things are today, and where you see them heading in terms of overcoming challenges and looking to the future?
Sure, and that's a great open-ended question that covers a lot of different aspects. So on the manufacturing side, so one of the key challenges with CAR T cells, at least the first iteration of CAR T cells, was it was one product for one patient. So it's all based on the patient's own T cells. That's challenging from a number of perspectives. Firstly, cost. So the cost of manufacturing for one patient a complex product like that, it's in the hundreds of thousands of dollars. And so the cost of treating one patient, is $300,000, $400,000, $500,000. And obviously, you can make that work for very rare diseases in the current situation. But once you want to try and treat diseases where there's lots of patients, so the solid tumors or something like endometriosis or whatever, where endometriosis is 1 in 6 or 1 in 7 of women of childbearing age. So it's huge numbers. So if you had to spend that much to treat a patient, just not, he would be on the healthcare system. So not achievable. So you need to get the cost of goods down, and you need to get to a point where you have one product for many patients, not one product for one patient. So that's the first challenge. Second challenge with the autologous CAR T products is that it takes best part of a month or so to manufacture, and so you got patients in generally pretty sick. They've failed existing treatments. You take their blood, you make the product. They got to wait a month to get the product back, and they will have deteriorated in that time. And then sometimes, the manufacturing process doesn't work. The patient generally has been through multiple rounds of chemotherapy. Their T cells are knocked around. They quite often older patients, so their T cells aren't that healthy anyway. And so the manufacturing process fails. And there is a significant failure rate for autologous CAR T products. And so you've got all those issues that you have to contend with an autologous product. us and a number of other companies have said, well, let's go with a stem cell-based product, give us an unlimited supply, be able to produce product, have it sitting in the freezer, ready to go directly into the patient as soon as they're ready to receive it. So no delay. The cost of manufacture should be much, much less. So the cost of goods for that product will be less, orders of magnitude less, and that's true. But these products are still very complex to make. So again, with our process, I mentioned before, we start with a gene-edited stem cell, an iPS cell. We have to expand those cells up. We have to differentiate them into a blood stem cell. Then we have to differentiate them into a sort of a precursor to the natural killer cell, and then differentiate them to the natural killer cell, and then expand them again. And so it's actually a multi-stage process. So it's not straightforward. And one of the challenges that we and others face is getting that process sort of industrialized, so that you can make large numbers of cells to an acceptable standard at a cost that will be acceptable in the field. So we put a lot of work into that. There's a lot of contract manufacturers doing likewise, and so we are seeing the emergence of a manufacturing capability for that type of product, and it will develop further. And as it does, the costs will come down, but it is still expensive. The reagents, the cell culture media, and so on are expensive. There are limited numbers of suppliers, so supply chain issues come into the equation, and so on. So it's not like it solves all the problems, but certainly it addresses a lot of the key problems of autologous products, and there are autologous natural killer products as well as T cell based products. So that's where we think the future is, and there are a number of other companies have come to the same conclusion. And I certainly believe that as we learn more about how to make the cells and to use the cells, and hopefully be able to use the cells at lower doses than what we currently need, because obviously if you have to go to really high doses of cells, then you need more manufacturing capacity. If you can work out how best to use cells, have cells that are more active rather than less active, and so on, get away with lower doses, then also the costs come down. So there's a lot of work still to be done there. So it's one of the challenges that face us. You asked about the future and where that might go. So I think there's a couple of different directions that people are going. There's the direction of taking cell therapies into different indications, and so effectively expanding the market, the application for that type of product. And I think that will come the more we learn about how to use the cells and when in the disease, and understand more about the biology of the cells and disease. So just to hark back to endometriosis, one of the things that we know is that in endometriosis, the patient's own natural killer cells are reduced in number and reduced in function. And so again, there seems to be a link, coming back to the bit of serendipity. There seems to be a biological link between natural killer cells and endometriosis, and so we would hope that if we then provide a natural killer cell, in terms of numbers and activity, then it will get round that deficiency. Now, that may or may not resolve the situation, but our hypothesis is that it will actually help. And so as more biology, as we and others learn more about the biology of the system, we'll learn more about exactly where our cells play, at what stage of disease, and so on. And I think there's lots of scope there. One of the other innovations in the field, people are moving towards the idea of actually doing in vivo CAR T cells. And so what they're doing is actually taking the viral vectors that introduce the CAR, the chimeric antigen receptor, to a T cell, packaging the messenger RNA up for the CAR in a lipid nanoparticle, and delivering that to the patient, and then effectively in vivo creating CAR T cells. That's much more like a traditional pharmaceutical product, and so we've actually seen a number of companies take an interest in that space. AstraZeneca just did a deal. I think they acquired one of the companies that is doing in vivo CAR T cells, and that's a way where you get around all the manufacturing costs. You make it much more like a traditional pharmaceutical. And again, it's one product for multiple patients. So it deals with a lot of the autologous problems. Challenge with that approach, though, is you are then reliant on the patient's own T cells. And so if you've got again, an older patient, a patient who's been through multiple rounds of chemotherapy, it may just be that their T cells are shot and, you're never going to be able to get them active enough to actually have an effect. So again, I think what we'll see in the field is that all these different approaches will advance, and ultimately it'll be a mix and match for different diseases, and quite possibly for different patients. It may be that an older patient comes in and you say, well, they're not suitable for in vivo. Autologous is too expensive. We'll give an off-the-shelf product to them, and even if the efficacy isn't equivalent, it may be that time and safety in that is the preferred way to go for that particular type of patient.
Yeah, it does seem that off-the-shelf cell therapies are going to be a big part of the future. And I'd like to know from Cartherics, what can we expect in the next couple of months, in the coming years, the next couple of years, particularly in terms of key milestones. I know that you mentioned getting into clinical trials. So can you talk to us about what we can look forward to from Cartherics?
Yeah, sure. So our lead product, CTH401, which is a CAR NK cell targeting TAG72, and has a couple of other gene knockouts that are associated with immune suppression. So we're hoping that particular NK cell will retain its function in what's called the tumor microenvironment. So in solid tumors, you have an environment which is basically hostile to immune cells. And one of the reasons that the checkpoint inhibitors work, they actually target one of the mechanisms by which tumors turn off immune cells from being functional. And there's a whole range of different factors in the tumor microenvironment that dampens down immune responses. And so we're hopeful that by making these gene edits, knocking out these immunosuppression genes, that our cells will be more functional in that particular environment. So that's our lead product. We've had 2 meetings with the FDA on that product, an interact meeting and a pre-IND meeting. And we are currently completing all of the work required to support an IND filing with the FDA. Our target for that is around the middle of calendar year 2026. So less than 12 months away for hopefully filing our IND. We've had excellent discussions with the FDA so far, so we're confident that we will have covered off all of the key issues for the FDA, and assuming that we've done that, then within a month after we file our IND, we will be able to commence the first clinical trial for CH401. So that will be in late stage ovarian cancer patients. We are currently we actually just opened a week ago our own clinical manufacturing facility. So we're actually gearing up to manufacture clinical batches of that product. So milestones coming up for us over the next 12 months. Clinical batches of CTH401, filing of the IND, starting first in human studies with CTH401. So they're all big milestones for the company, and all first. It will be the first time Cartherics has manufactured product for the clinic. First time it's ever filed an IND. First time it's ever done a clinical trial. So that will be great milestones for us as a company. We have another product that is actually an autologous CAR T product, has the same gene edits as our CAR NK product, and we actually used effectively donor-derived T cells to validate the gene edits that we did for the CAR NK cells. And that product, we have a partnership with Peter MacCallum Cancer Centre here in Melbourne to take into the clinic, and we have a Chinese partner, Shunji Holding, who are developing it in China for the clinic. And so that product we hope will also go into the clinic in about the next 12 to 18 month period. And so that will be a significant milestone. Not that we were the company taking it into the clinic. For us, it was a distraction, but it's based on Cartherics technology. And like I say, it has the same gene edits that in our CAR-NK product. And so that will be a milestone for the company when it comes up. And then we have a couple of products that we have in earlier stage development targeting tissue factor. So our second tumor and endometriosis target. And so we will be aiming to get those products into formal non-clinical development during the course of 2026. So probably by the end of 2026, we'd aim to have effectively a package that we go and have a pre-IND meeting with the FDA around one or both of those two products. One is a simpler product focused on endometriosis. The other is a more complex product focused on cancer. Again, both targeting tissue factor, but the one on cancer has more modifications, again, with the view of trying to make the cells more functional in that tumor microenvironment. And so, yeah, we'd hope to be in a position to have a pre-IND meeting with the FDA towards the end of 2026 or early 2027 around one or both of those products. And again, that will be a significant milestone for the company. And along the way, we'll generate additional supporting data, both in cancers, a range of cancers, including triple negative breast cancer, where again tissue factor is a target, and data in endometriosis. So again, supporting that biological connection between ovarian cancer and endometriosis. So they're all the things that we're planning to do over the course of the next 12 to 18 months. The other big thing for us is raising money. It's an expensive exercise, and so we will be out there. Well, we've just started the process of the next capital raising for the company, and so we'll be out there looking to bring on board a new group of investors, plus probably some of our existing investors. And so in the next 12 months or so, we would expect a major announcement around financing for the company.
Wonderful. A lot of firsts, like you said, and a lot of amazing, flurry of activity definitely to look forward to in the next year or so. And we wish you all the best with all of those milestones. And we'll definitely be keeping an eye out for all of them here at Xtalks. And yeah, thank you so much for all of the work that you do, for your time. And I just want to end off with a lightning rapid fire kind of a round, just to leave off on a it's already a really high note, but I just wanted to get your insights in terms of, quick one word, one sentence kind of answers. So hopefully you're ready for this. Okay.
Okay. My forte is not one word answers, but I'll try.
No, I didn't mean to say anything like that, but no. Yeah, me neither. So we'll try this. We'll see how it goes. But okay, so the first question I have here is, I know that you've talked about some of these things already, but maybe if you could just wrap it up in a word or a sentence. One innovation in cell therapy that excites you most right now.
Well, it's difficult to limit it to one, but it's in vivo CAR T, I think, is an interesting prospect. I think cell therapy broadly in non-oncology diseases, I think, is a huge the way of the future. And I'll just put in a plug for a book here, because it's a great book. So there's an oncologist by the name of Siddhartha Mukherjee. He's a Pulitzer Prize winning author, written a number of books. There's a book he wrote a couple of years ago called The Song of the Cell. People should read that. It just reminded me of just how important the cell is. He makes the point: cell is it's the core of all physiology, and it's a core of all pathology. And once you read that book, you just realize just how critical the cell is in everything, and how cell-based therapies, the potential for them is just so enormous. So I recommend anyone who hasn't read that book to read it, because it is a great read across all the different aspects of cells and just how powerful cell-based therapies can be.
Wonderful. And a leadership lesson that you've carried with you throughout your career?
Gee. So I'd say, um, from my time at Millennium, Millennium was a phenomenal company. So it got acquired by Takeda back in 2008. But it was just an amazing company. It founded in 93. So, it was in existence for 15 years, but it was just a hothouse of innovation, the smartest people. And it was just a buzz all the time. Just an amazing place. But the leadership thing, Mark Levin was the CEO. He founded the company. He was the CEO. Mark is my gold standard as a CEO. And the thing that Mark taught me was humility. Surround yourself with really smart people, and you don't have to have all the answers, but you need to ask all the right questions. And Mark was just phenomenal. He surrounded himself with the smartest people on the planet. Um, but in any meeting, he was the one that just asked the right questions, and no ego about it, but just ask the question. So yeah, that's what I learned from Mark. From Mark, I I'm no Mark Levin. I try and emulate him where I can. But just an incredible leader, and someone, um, that his staff, they would walk over red hot coals for him. It was he was just a fantastic leader, and he built the Millennium community, and that community now is out there in hundreds of different biotech companies. It's just fantastic work, and I'm proud and very privileged to have been part of that community.
I love that. Leading by example and with humility and asking the right questions. I think that transcends any and all fields even. So that's universal advice.
Absolutely,
Yeah. Wonderful.
absolutely.
And so what powers your mornings? Coffee, tea, or a great idea or something else?
So
It's a random one.
coffee is yeah, coffee is the starting point. So, and this comes from in my days at Millennium, carried through to my days at Cartherics. So, first thing in the morning, have a coffee with at Millennium with one colleague and my best friend there, and just kick around ideas, chew the fat, talk about the world or talk about work. That's carried through to Millennium as the starting point for the day, and it's a nice way to start the day, and you know, it's a good way to just remove yourself from the strict work environment for a little while. And yeah, sometimes you talk about work stuff, sometimes not, but yeah, that's pretty much my go-to for the start of the day.
Great. And lastly, one word or maybe a couple I'll give you to describe the future of CAR NK therapy in particular.
Look, I think it is enormous. There is, we're just at the start of this field. It's been around, a decade. Well, cell-based therapies, people started working with them in the late '90s, but they really didn't come to the fore, immune cell therapies, until 10, 10 or 15 years ago. So we're right at the start of that field. I think the potential is enormous. It's going to take time and money and persistence, to work out how best to use them. And all those things are critical. And I've been around long enough. I've seen this multiple times. I saw it with antibodies, with antibody drug conjugates, with gene therapies. Pretty much any innovation. From start to when people really understand how to work with these things. We're talking decades, minimum. 20 years, and longer to really work out how to properly use these new sorts of treatments. And so we're still at the start. So I think the future is enormous, but it requires companies like us to be persistent. It requires investors, with patient investors who are prepared to put their money in for the long term, and it requires grant ultimately from partners.
I love that. The future is enormous. And thank you, Dr. Nisbet, for really being at the forefront of leading some of this enormous work. And we wish you all the best. And we're definitely going to be keeping an eye out here at Xtalks for all the wonderful milestones coming up from Cartherics. And thank you for everything that you do. And thank you for sharing all of those insights with us today.
That's been my pleasure, Aisha. Thank you very much.
Thank you. All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.